Okay, so now that we've talked about the mechanics of the action potential, one of the big questions that I have is, why does this matter? And so what we're going to talk about now is the spread of this action potential. And I hope by the end of this you will understand why does the opening and closing of these different channels, why does it matter? What does it mean? Okay. So just to remind you, we have talked about resting potential, graded potential, and action potential. Now we've just talked about the start of it. Action potential starts here in the axon hillock region. But then action potential must spread down the entire axon. We call the spread of action potential propagation. Okay, so that's this word right here. Okay, so what I'm going to do is I'm going to switch to our active learning exercise cartoon cutouts and show you how this action potential spreads, and then we'll move on to a couple of different other things that hopefully help this concept make sense. Okay, so now what I'd like to talk about is I'd like to remind you of these cartoon figures for the action potential. Okay, so if you notice here, we've got our cell membrane represented by this black line. This is outside of our cell, inside of our cell. You should be familiar with all of this from the last video. Okay, we have a high concentration of sodium over here. We've got a high concentration of potassium on the inside of the cell membrane, and we have these channels that you've been familiar with before. And finally, a positive membrane potential outside and a negative membrane potential inside of the cell. Okay, as we've discussed before, neurotransmitter binds to the chemical gated sodium channel, and that will open it. And that allows a little bit of sodium into the cell. Now, I just want to remind you that this channel is located in the cell body and the axon hillock. So initially, this sodium is going to go to the voltage-gated sodium channel that is located at the initial segment of the axon. This channel, once there's enough sodium to reach -60, will open, and at that point, a rush of sodium will enter the cell. So a reminder, this switches the membrane potential to positive on the inside and negative on the outside. Okay, well now what we're looking at is how does this start of the action potential, it starts with the opening of this voltage-gated channel. How does that then travel down the entire axon? The entire cell membrane of the axon is moving this way, as shown by the arrow here. Well, it turns out if you continue to look down this membrane, there's going to be another set of channels embedded in it. Okay? And what happens is a little bit of this sodium just diffuses over right here. And at this point, this is when this channel, if there's enough sodium that diffuses over and it reaches -60 at this place in the membrane, then this channel will open. And that starts an action potential right next door. And so we get the same series of events. Sodium rushes into the cell. Okay. Now I do want to make the point is as this is starting, this behind is finishing up. So we will have this channel open, the potassium rush out, the channels reset, and the pump will bring things back to their normal locations. This membrane potential also flips back. But this right here continues to move as it did. So first, a little bit of sodium diffuses over to its neighbor. Now, even if some of the sodium diffuses back, this stuff behind is still in the process of resetting, so you can't trigger an action potential going backwards. You can only trigger it going forward. So, a little bit of sodium diffuses, we reach threshold. Here we've got an open channel and a rush of sodium comes in. Now behind, all the events that are normal are still going on. This is opening, the rush of potassium goes out, the channels are resetting, and then the pump resets these ions. Okay. Again, a little bit of sodium would diffuse over here, trigger channels that would be located in front of it. Action potential happens here. So this movement, action potential, started up here in the initial segment, and it will move along the entire axon because these channels are embedded along the entire axon. Okay, and so now I'd like to give you another visual representation of this same concept of propagation. I'm trying to give you a number of different ways of viewing this because hopefully one of these ways will click for you. So the first thing that we have here is I just want to show you that this is representing the cell membrane right here. So notice these are your phospholipids that you saw back in chapter 2. Okay, and so what we've got here is just a simplified representation of what I just showed you. So at each location along the axon, so this is the axon membrane, we've got closed channels. So everything starts off as closed prior to the action potential beginning. Now, all we're going to look at is the charge across the membrane, the positive outside and the negative inside. Okay, so let's take this in another step. Channels open. Say this is the initial segment right here. Sodium rushes in, and we get a movement of the inside to become positive, and the outside is negative. This is going to trigger the gate or the channels in front of them to open. So we get an open, and again, a change across the membrane right in front of the initial segment. Now at the same time, this part behind, the channels are closing and it's resetting the charge. But this one in front has been triggered to open, and we get a change across the membrane. The one behind resets the channels and resets the charge. And so this continues on down. We get this positive charge moving to the inside of the membrane down across this way. And so if we just want to look at where is the positive charge on the inside, it starts here and it moves down to the end of the axon. And if you know anything about what the movement of a charged particle is, it is called electric current. So it is literally electricity running through your neurons. Literally. So I'd like to give you a brief video snippet showing this. So first of all, here is going to be a close-up of your brain. So here are neuron cells, and that bright light is the electricity that's running between one neuron and another. Now we're getting up close to the axon, and something's moving in and out along the length of it. And And you notice it is that positive charge moving in and out, in and out. And that is literally electricity moving until it reaches the very end of the cell. Some students also find it helpful to think of this as dominoes, and here is a video embedded in your homework showing this. It's this huge domino set, and you can think of these as channels being set right next to each other all along the axon. And so here we go. And this looks like it took quite a deal of time to arrange and set up. So now we're ready to go. Initial segment being triggered, an action potential moving down the axon. And so this is something that helps me think. This is why it's not easy, or it's not possible to do it the other way, because you've got to pick it up and reset it before you can do it again. One last thing that I would encourage you to do is that on the very last page of your lesson is a segment about why this matters, and it brings up a video that has this woman, this newscaster, explaining how researchers are actually using, tapping into the electricity in your neurons to actually interface between your thoughts and a computer screen. And this is being used to treat people that have ALS, Lou Gehrig's disease, that inside their thoughts are fine, but they've lost the ability to have any sort of motor movement, so they can't express those thoughts. So can a computer form that missing link? So I encourage you to look for this video on the last page of your lesson.